about 50 years ago, I wondered why medicine was all about anatomy, when in fact Newton told us that to have any physical structure to be animated, it needed energy. And so why weren't we talking about the energy of medicine? I like to tell the medical students it's all fine to dissect a cadaver and learn everything there is about the anatomy, but if the patient leaves your office in the state of the cadaver, you've made a mistake because the patient's dead. And so wouldn't you want to know more about why the patient is not dead than why you know about the anatomy? So we thought, well, maybe if we put an energy in the middle of medicine instead of anatomy, we might have new insights into the common diseases and maybe even the rare diseases. So, of course, at that time, it was clear that the mitochondria generated most of the energy in an adult or any living human. And that oxidative phosphorylation is how the mitochondria makes energy. It generates energy, it makes reactive oxygen species, it regulates the redox potential, and it regulates calcium. And so how does the mitochondria generate this kind of elaborate system? Well, it requires nuclear genes, and those genes could be mutated, and they could give mitochondrial disease. It has to regulate those genes through the epigenome. It could have changes in the mitochondrial DNA, the maternal inherited DNA, and that could be some ancient polymorphisms or recent deleterious mutations, or it could be affected by your diet, or whether you exercise, or if you smoke and you poison your mitochondria. All of those reasons are environmental reasons that could impact energy. And if you damage the mitochondrial energy generating system, you increase reactive oxygen species, more calcium is taken up into the mitochondria. That damages the mitochondrial DNA, and there are thousands of copies of the mitochondrial DNA in each cell. So the more you damage the mitochondrial DNA, the less energy there is, because the mitochondrial DNA is basically the wiring diagram of the power plant. And so you would get an age-related decline in mitochondrial function, and when it crossed the minimum for the tissue to function, you would get a symptom and a disease. So it seemed to me obvious that if we studied energy, we might have some interesting insights into common diseases. So what are the systems that might be most affected by energy? Well, the brain is 2% of your body weight, uses 20% of all your body's energy. So wouldn't it be illogical that a slight change in mitochondrial energy would affect your neuropsychiatric problems? But then as you've heard, the heart, the muscle, the endocrine system, renal system, all are very high energy demand tissues. So those are the ones that would be affected by energy deficiency. And what are the ones affected by common disease? The brain, heart, the muscle, and endocrine system. So that seemed logical. But what happens if energy is impaired, if the mitochondria is impaired? Well, it burns with the substrates, fatty acids and carbohydrates, with the oxygen you breathe to make energy. So if there's a defect in energy production, then you're going to have carbohydrates and fats build up, and that's metabolic syndrome and obesity and all those areas. And the mitochondria is basically a bacterium. So if the bacterium is clearly going to be regulating how the immune system is going to interact, then we would affect changes in the mitochondria might affect the immune system, which would be called the inflammasome, CS, like receptors. So then suddenly it made sense why all the common diseases are also inflammatory, because of that. They're interacting with the same mitochondrial energetic effect. So that then led, so how is this all concept going to tell us something about mitochondrial transplantation? Well, I like to think of the problem as two different kinds of problems. The first problem is if you want to change, say, the mitochondrial DNA and make it better, then you have to get the mitochondrial DNA in the cell, and it needs to persist. So what would you need? Well, first of all, you need to have many mitochondria changed. Second of all, it might be very important that the mitochondrial DNAs are species specific, so you might worry about the species. You want to have a permanent change. You would like to have a cellular change. And there are ways to deliver mitochondria. One we developed in 1971 is so-called the cytoplasm or cyber transfer, where we actually take a cell, the nucleus infused in cytoplasm, which is a bag of mitochondria, and transfer it to the cell. But you could also think about mitochondrial transplantation, or you could be impressed by Dr. Wilson's wonderful talk and just use mitochondrial DNA. So if you had then these genetic changes, you would have a genetic process that's altered, and you would then have to have replicating donor mitochondrial DNA for a long time effect. But there might be something else you might want to do. You might want to just change the phenotype. So that may not require a genetic change. So you would then maybe only need a few mitochondria. those mitochondria would not be species-specific or strain-specific. They would be important for tissues and organism change. And they would not necessarily be involved directly in cells, but they might relate to changes in tissues and changes in animal function. So then you're looking at animal physiology. Well, this gets back to this idea that we had, that maybe mitochondrial energetics has multiple different effects. So maybe it's going to affect the interaction of metabolites with the epigenome, maybe it's going to affect the immune modulation, or maybe as the mitochondria makes reactive oxygen species, it might affect antioxidant defenses. But what's interesting is you can imagine then a permanent change in the mitochondrial DNA would also change this other system. But the converse is not true. A transient change is not going to change the genotype. So with that idea then, maybe we should think about these two as separate ideas. So starting with the genetic idea and changing the mitochondrial DNA and looking toward Wilson's approach. So a number of years ago, 54 years ago, we developed a method we call the cyber transfer, where we took a cell. At that time, we had isolated cells that were resistant to a mitochondrial ribosome inhibitor called chloramphenicol, and that changes the mitochondrial ribosomal gene. So you can select from mitochondrial DNA for the first time, and that allows us to work out the genetics of the mitochondria. So you take the chloramphenicol-resistant cell, you remove its nucleus, you take the cytoplasmic fragment, fuse it to a chloramphenicol-sensitive cell, select for chloramphenicol, and if it was cytoplasmic inherited, it transfers, and it did, and that was good for me. But there's also then fusion of two different cells, and we call that a somatocell hybrid. So then you fuse the two cells, the two nuclei come together, and now you have a mixed nuclear and cytoplasm. So we thought, well, what would happen if we transferred chloramphenicol resistance between cells? So we enucleated, that's what I have here, a mouse cell, which is chloramphenicol, and fused it to another mouse cell, and we got lots of colonies. Great, that worked. We took enucleated human chloramphenicol resistance, fused it to human cells, got lots of colonies. But then when we enucleated the mouse chloramphenicol cell and fused it to human cell, got no colonies. And we took a chloramphenicol resistant human cell and fused it to the mouse cell, no colonies. So what that says is intraspecific works, interspecific doesn't. And then we made a somatic cell hybrid between human and mouse. And traditionally, we know, or we found, when you make a somatic cell hybrid, the first thing you lose is one of the mitochondrial DNAs. And in almost all mouse-human hybrids, it's the human mitochondrial DNA. And when you do that, you retain the mouse chromosomes, but you lose the human chromosomes. So the chromosomes go along with the mitochondria. So I said, well, what if we select for chloramphenicol-resistant human mitochondrial DNA? Well, that made cells really, really sick, but ultimately we got some colonies, and what they did is they retained the human mitochondrial DNA and the human chromosomes and lost the mouse. So what does that say? That says the interaction between the mouse and the mitochondrial DNA and the chromosomal DNA is really, really important. Okay. So then we said, well, what if we take different strains of human mitochondrial DNA? So we used a cell line called HD1080 and another cell line called HeLa, and they're of different origins. So if you take HeLa chloramphenicol resistant cells and fuse it to an HD1080 cell, you get lots of colonies and they're very stable. But what happened when we took the HD1080 mitochondria, chloramphenicol mitochondria, and fused it to a HeLa cell, initially you got lots of colonies, but then they began to all blow up and they all died. So it wasn't absolutely true that all the intraspecific mitochondria worked. Sometimes they did, and sometimes they didn't. And what was interesting then is this post-effect of the colony formation, which was, we now know, apoptosis. So it really does matter what the mitochondrial origin is, if you want to establish the mitochondrial DNA permanently. So what are some of the variations that might be relevant? Well, one of them is there's ancient variation that accumulated as our ancestors arose in Africa and migrated around the world and gave rise to what we call different mitochondrial haplogroups or lineages, and we gave them different letters. But what's interesting is these turn out to be adaptive lineages. So when people came out of Africa, they acquired a mitochondrial DNA mutation that allowed them to adapt to cold. And then they acquired additional mutations that allowed different sub-regional adaptations, some of which are immunological. And these are very important because they relate to cancer predisposition and other kinds of phenotypes. So then that says that maybe the haplogroup changes weren't so important if you just want to change the phenotype, but they might be very important if you want to change the genotype. And so then we have to think differently. So then we said, well, what about changes of the phenotype? So we're now back to a few mitochondria changing, then interspecific changes if you need, and so on. Okay, so the one thing we did was, what would be the factors that are important in a few mitochondria changing the phenotype? Well, we might think of several factors. One, the mitochondrial DNA might change the epigenome, or the mitochondrial DNA might change the mitochondrial biogenesis, or the mitochondrial DNA may change the reactive oxygen species, or the mitochondrial DNA changes might change the inflammatory system. So let's look at all of this. So first we took cells, the same nucleus with different percentages of the MELOS mutation, which you've heard from one of our families. And we had different percentages of MELOS mutation with the same nucleus. And then we did RNA-seq, and what we found is a very striking result. One of the things about the MELOS mutation is if you have 10 to 30% mutant, you get type 1, type 2 diabetes or autism. If you have 50 to 90%, you have stroke-like episodes, cardiomyopathy, and muscle disease. If you have 100% mutant, you die as a child with Lee syndrome. So how could you have such different phenotypes with exactly the same mitochondrial DNA mutation just by a quantitative difference? And it turns out that that quantitative difference regulates the epigenome. So in fact, if this is a cell with normal mitochondrial DNA, this is its transcription profile. If this is the cell with 10 to 20% mutant, this is autism, this is neurodegenerative disease, this is lethal Lee syndrome. So this mitochondria regulates the epigenome, and it does that by the mitochondrial DNA changing the electron transport chain, which changes the NADH to NAD ratio, and as you just heard, that will change the TCA cycle, and all the TCA cycle intermediates are the intermediates to drive the epigenome. So what we found is 150 epigenomic changes that determine the nuclear gene regulation based on the mitochondrial genotype. So a small change in mitochondrial function could have a huge effect on gene expression and therefore the phenotype. We then made mice where it had two different normal mitochondrial DNAs, and then they segregated, so this is maternal inheritance of these two different mitochondrial DNAs. If we take a mouse that is what we call heteroplasmic, and we then make a mouse that has one mitochondrial DNA, 129, another NCB, and then we ask, are they different? When we ask the mice if they learn a memory test, this is a Barnes maze, and these are different holes, and under here is a little black box for the mouse to hide in. So if you put a mouse there, it goes around, finally it learns how to find that hole over several trials. And that's fine. All mice did that great. But then if you wait 2 days and put the mice back on, then the homoplasmic mice with one mitochondrial DNA find the hole, the homoplasmic mice with the other mitochondrial DNA find the hole, but the heteroplasmic mouse has completely no long-term memory. It's just wiped out. So that subtle changes on haplogroup has a huge effect on phenotype. So we can then actually ask these different mitochondrial DNAs, BLAC6 and NCB, and if you have an injected tumor in the NCB, it completely blocks the tumor, just the mitochondrial DNA. But if you have BLAC6 mitochondrial DNA, tumor grows rapidly. And if you simply change the mitochondrial ROS production of the hemopoietic stem cells, then you can actually alter this tumor rejection and also transplantation. So again, subtle changes in mitochondrial function have huge effects on phenotype. So the other area is the environment, and so we worked on SARS-CoV-2. And SARS-CoV-2 proteins, and 19% of the cellular interactions of SARS-CoV-2 proteins actually block mitochondrial function. Why would it do that? Because it inhibits mitochondrial respiration, and as you just heard from Hazel, that then drives all the metabolism over to make substrates for biogenesis, and the virus wants to make biogenesis, not support you. So what it does is it inhibits mitochondrial nuclear gene expression to block mitochondrial function, drive glycolysis, and make substrates. And so if you look at a person who's died of SARS-CoV-2 and you look at the expression of all the nuclear encoded OXPHOS genes, and brown, black, blue is off, every one of the OXPHOS complexes is completely shut down. So that, in fact, is the reason why long COVID is so toxic, because, in fact, long-term shutdown mitochondrial function. And the way it does that is it has a particular protein that goes to the nucleus and it completely shuts off oxidative phosphorylation. I'm almost done. So what we can do is take then a nuclear encoded catalytic antioxidant and put it in the mitochondria. We can do this two ways. We can use an enzyme catalase that takes hydrogen peroxide to water and hydrogen peroxide is found in the mitochondria. And when we do that, in fact, this is the untreated mouse with the virus, and this is the treated mouse. It becomes resistant. And we also have a drug, UK, and it can give you exactly the same thing. And what does it do? It completely reverses the inhibition of the mitochondria and the inflammatory system. So they're all linked together, and they're all giving you this complex phenotype. So what we really then have is a system that is sensitive to signals. Now, what are the signals? They could be metabolic, they could be inflammatory, they could be photons, all of these areas that we've been thinking about, how this could happen. But what happens when you have substrates and you go to the mitochondria? If you have good mitochondria, you burn the substrates and you make energy. And if you have bad mitochondria, you make ROS, activate HIF1 alpha, that drives you into forming substrates. And so the virus wants to go this way. But early development is exactly that problem. You have to use your substrates to make cells. So you're in fact glycolytic and living in an anaerobic environment. So these are all the same for cancer, for development, and for virus. So we can then take antioxidants, we can change this procedure and drive it this way, but there's a problem because the antioxidants affect the inflammatory system. And that'd be very good if you want to damp out inflammatory, but it's very bad because the inflammatory system regulates nascent tumor formation. So if you in fact wipe out your antioxidant defense, and you use antioxidant defenses to wipe out your immune system, you would then get a new problem, which is developing tumors. So we have to be very thoughtful about what we do with these systems, and these are the people that do the great work. And this says, stop.
Thank you, Dr. Wallace. So we have 1 minute for questions. If there's any question.
Okay, I'm here. So there is a recent publication on science immunity where they have spoken on SLE, systemic lupus erythematosus, where they have exposed, I think on mice, and they also have done experiments on human, where due to UV exposure, the mitochondria gets fragmented and then release ZDA. And then the ZBP is the protein which comes into play. And then the C-casting pathway gets upregulated. So do you think that this mitochondrial transplant can be a good thing for the SLE? Because
I think Dr. Becker did a fabulous job of telling us where we are now and what we need to do. That's a great hypothesis, as are any of these. They're hypotheses. We don't know yet. What's so exciting is we're here to find out. And I think there's so many exciting things to do. But one thing we have to be very careful about. I'm a clinical geneticist. I care about patients. And we have to solve the patient's problem. But it isn't necessarily simple, because what might be good short-term might be very bad long-term. And that's one of the messages I want to show. So we need to understand this well enough that we're sure that we don't just solve the problem transiently and then make it worse. And that's what our clinics are dedicated to do, long-term health of our patients.